Hello again, and welcome back to yet another narrated lecture. In this lecture, we're going to be discussing Chapter 11, Cell Communication. You can use this narrated lecture in conjunction with the lecture review sheets for Chapter 11, and/or the printable versions of the slides that are available on Blackboard. All right, in this chapter, we're going to be discussing cell communication. Now, the ability to communicate with the environment and with other cells is an essential characteristic. It's an essential skill that cells have to master. They have to be able to do this in order to stay alive. Cells that live on their own have to be able to communicate with their environment so that they know what's going on. Cells that live in a multicellular environment, that live in colonies or live inside an organ or a tissue, well, they have to be able to communicate so they can work together as an organ. So for instance, the cells inside your liver have to be able to talk to one another in order for the liver to function as a liver. So it's a really essential skill. Now, when cells communicate with one another, they have a language that's a little bit different than what we have when we're speaking to one another. When we speak to one another, what we do is we produce sound waves with our throats and with our mouths, and those vibrations go through the air and eventually they hit someone's ear. And they vibrate the parts inside the ear, and the brain interprets those vibrations as some kind of message. Cells do something kind of similar, except they use chemicals instead of sound waves. So here we have an image that sort of shows that. We have the membrane of one cell here. Here's the membrane of one cell. And here's the membrane of another cell way over here in the distance. And what you can see is that this first cell is releasing these chemicals into the extracellular space between the cells. And that chemical is a message. Different chemicals have different meanings depending on which ones are released, in which combination they are released, and the timing of when they are released. So this really is a molecular language that cells are able to speak. Now we're going to discuss several different types of communication that cells use, but all of these forms of communication involve these two types of structures, receptors and ligands. This molecule is a receptor. This receptor is a type of protein. Now this molecule over here is known as a ligand or a ligand, if you want to say it that way. And so this is the message that's actually being sent from another cell. And so what will happen inside these systems is that the ligand will bind with the receptor, and they fit together like a lock and key, like an enzyme fits together with its substrate. So this is how cells receive very specific messages from other cells and/or from the environment. Now, because this receptor is a protein, it is able to change its shape. It's able to change its conformation, and that can elicit a response inside the cell. Now, there are many different forms of communication, and there are a couple of different ways that these can be classified. So we're going to talk about two classification systems for these methods. The first way to classify these communication methods is based on the location of the receptor and the location of the ligand. Some ligands and receptors are located inside the cell, so we call them intracellular. So for instance, in this diagram, you can see that the receptor is actually inside the cell here. It's in the intracellular environment. Now, in order for the ligand, which is here, to bind with it, it actually has to go all the way through the cell membrane. Here's the membrane. So it has to be capable of sneaking through that membrane by itself. Now let's think about the types of molecules that can do that. Back in chapter 7, you learned that molecules that are very small and are uncharged or nonpolar, those are the ones that are able to sneak through that membrane by themselves. A great example of this type of ligand are the steroid hormones, including such things as estrogen and testosterone. Those chemicals are very nonpolar. They are lipids. So they are able to go through that membrane by themselves, bind with the receptor, which is intracellular, and then that receptor will change its shape and it will elicit some kind of response. In this case, it's interacting with the DNA and it's causing some kind of specific protein to be made. Okay, but that's how testosterone and estrogen act, is by going inside your cells. They are hydrophobic molecules. Now it makes sense that if a receptor isn't inside the cell, then it's probably outside the cell. So these types of receptors and ligands are known as extracellular, and that means that the receptor itself is located on the membrane. Here we have a receptor, change that color, and here we have the membrane itself in yellow here. So it's on the outside of the cell. The ligand in this case is not going to be able to cross that membrane. It's either going to have a charge or it's going to be very, very large. Something like that is going to prevent it from going inside the cell. So its receptor has to be on the outside of the cell. Now, if it's on the outside of the cell, how does the message reach the response inside, cause that response? Well, when the ligand binds with the receptor will change its shape, its conformation, and that will cause this whole giant series of chain reactions to happen. One molecule will affect the next, and so on until a response is finally caused at the end. That kind of a pathway is known as signal transduction, and we're going to talk about that quite a bit more later. All right. Now, when you finish this narrated lecture, there is going to be a quiz. I've created the quiz in Google Forms, and I'll tell you how to access it later. But for now, I want you to pay attention to these questions as they come up. You will be asked to respond to these questions, and you are going to have to analyze the information in this lecture in order to get the correct answers. So here's your first two questions, and they have to do with either intracellular or extracellular receptors. All right. So the second way we can classify types of communication is based on the distance over which the message is sent. Some messages only have to travel a little ways, and some messages have to travel a great distance between cells. So first we have this type of signaling called direct signaling. Direct signaling occurs when cells are actually physically touching one another. So there's no distance between them. White blood cells are a great example of direct signaling. Here we have a white blood cell, and what these guys do is they patrol your body, and they are looking for invaders, bacteria and viruses that are not supposed to be there. And the way they sort of check the IDs of the cells and other molecules they run into is they will come down and they actually bind with receptors on those cells. So let's say this is a liver cell, and this white blood cell is checking it out, making sure it's healthy. It will come down and bind with its receptors, and that tells it whether it belongs there or not. So they're directly touching, direct signaling. There we go. Now, these little structures called gap junctions actually connect the two cells together. So the cytoplasm of one cell has access to the cytoplasm of another cell, and signaling molecules can be directly passed between the two cells. These gap junctions are made of proteins that can twist to open, like that, or twist and close. And so that helps the two cells control what comes in and what goes out. A great example of an organ with lots of gap junctions are heart cells. Think about it. Heart cells have to work very carefully together in order to give the heart the right rhythm. So they need to be in close contact with each other and communicating very well. And so heart cells have lots of gap junctions that help connect them. Now over here we have plant cells. Plant cells have cell walls. So cell walls are thick and they are a barrier. And so in order to pass things back and forth between plant cells, they will have these little tunnels built in. And we can't just call them tunnels, we have to call them plasmodesmata, right? Big, long, crazy word. But these little tunnels that connect the cells allow things to flow from the cytoplasm of one to the cytoplasm of another. It helps them communicate and pass materials back and forth. So that's direct signaling. The second kind of signaling based on distance is called local signaling. There's two types, which we'll talk about. But local signaling occurs over a short distance between cells. So they're not touching. They're separated by a little distance, but not a very big one. First type of local signaling is known as paracrine signaling, and you can see it in the diagram here. In paracrine signaling, a cell, such as this one, will send out chemicals. Here's the messengers, the ligands, and these will travel across the space. So this is the extracellular fluid out here, and they will eventually bind with receptors on the target cell. So this is a pretty efficient way of communicating because one cell that sends out a bunch of signal can talk to all of its neighbors all at the same time. Another type of local signaling is known as synaptic signaling. This type of signaling only occurs between nerve cells and whatever they are connected to. That being other nerve cells or your other organs, like your muscles or your guts. Look at the shape of these weird nerve cells. They're almost star-shaped. Instead of being nice big fat round cells, as we would expect, neurons, these nerve cells, have these long crazy extensions that stick out. And these extensions allow them to make connections with other cells. There we go. Other nerve cells or your organs. Now at the ends of these long extensions, you're going to find these regions known as synapses. And that's what this type of signaling is named for, these synapses. So let's take a closer look at those. This is what a synapse kind of looks like. You can see that this is the end of that nerve cell. Here it is. End of the nerve cell. And it kind of expands into a little button-like structure. In fact, one of its names is a bouton, which is a fun name for it. Now what this thing is going to do is it's going to release chemicals into the space, and the space itself is known as the synapse. But these chemicals flood into this little space and they bind with little receptors on the target cell and tell it to do something. Now sometimes the message doesn't get efficiently sent. You may have heard of a medication known as an SRI or an SSRI. And what that stands for is a serotonin reuptake inhibitor. So what's that all about? Well, in certain types of depression, what's happening is that neurons in the brain are releasing serotonin into this space, but the serotonin isn't staying in there very long. It's getting sucked back up into the nerve cell really quickly. The reuptake is too fast. And so because it gets sucked back up by the nerve cell, it's not sending the proper signal. It's not a very loud signal. So what SRIs do is they sort of block that process. They prevent the neuron from taking that serotonin back up quite as quickly. So it stays in the gap a little bit longer and it sends a little bit louder signal. And that seems to help reduce some of those depressive symptoms. Chemicals that are released inside synapses are known as neurotransmitters. And so there are many chemicals in this category, including serotonin, melatonin, norepinephrine, epinephrine, and many, many others that you'll learn all about if you ever take an A&P course. All right. Now the next type of signaling is known as endocrine signaling. This is sending a signal over a very long distance. When organs are separated by a great distance in the body, they can still talk to each other. This type of signaling takes advantage of the circulatory system. So you've got this system of blood vessels that spreads all through your body, connects to almost every organ. And so what these cells will do is they will release chemicals into the blood. That signal will travel through the blood all the way to a target cell. And it will bind with a receptor on a target cell and tell it to do something. So the key to endocrine signaling is the use of blood and the use of hormones. These chemicals are called hormones. Now you have an organ system inside your body known as an endocrine system. So what's that about? Well, as you can see from this diagram over here, the endocrine system is a series of organs spread throughout the body, and all of these organs release hormones. So it includes things like the thyroid in the throat, the thymus that sits on top of the heart, parts of the liver can act as endocrine organs, the pancreas, ovaries and testes can also act as endocrine organs. All of these organs release hormones into the blood at different times and different amounts to send different messages. This slide is just in here to remind you or to point out that chemicals that are classified as neurotransmitters may also act as hormones in the body. So what's the difference? Well, really the difference is where it's released. A neurotransmitter is released inside a synapse between nerve cells. A hormone can be the same chemical, but if it's released in the blood, we call it a hormone, not a neurotransmitter. So for instance, epinephrine can act as both a neurotransmitter if it's released in the brain, or a hormone if it's released in the blood. All right, this refers you back to your quiz. At this point, you're now ready to answer these questions. And these questions are going to ask you to examine these diagrams and decide which diagram represents which type of signaling. Should be pretty easy to figure out. Once you've completed those questions, you can do the next 4. These questions give you some scenarios and ask you which type of signaling is involved in each. So for these, you have to look carefully for clues. But again, that's part of the quiz. All right. So in part 1 of this lecture, we were talking about different types of cell communication. In part 2 of this lecture, we're going to discuss something called signal transduction in a little more detail. And I'm going to give you some examples of some common signal transduction pathways. So the definition is written there. You can read that. Let's talk about how these pathways work. All right. So these pathways can be quite complex. Here is a simplified diagram of that type of pathway. All of these pathways begin with a ligand and a receptor. So a ligand is going to bind with a receptor, and we call that step reception. That receptor is going to change its conformation, its shape, and that's going to activate some other molecules. Sometimes that just activates one molecule, and that activates another one, and that activates another one, kind of in a row. Sometimes the activation of the first molecule can activate many different other types of molecules. So a whole team of molecules is activated in this step. And so you can get a really big response at the end from just a single signaling molecule. But this second step here, where the message is being passed further and further and further into the cell, is known as transduction. And this is also the step at which the message can be amplified. One little whisper outside the cell can result in a loud shout inside the cell because the message gets amplified. Finally, the last step of these pathways is known as the response. So that's when the cell responds. It does something. This can include turning a gene on or off, turning enzymes on or off, or maybe even releasing something from the cell, or taking something in by the cell. Any of those things can count as responses. And they're all hooked to specific signals and specific pathways. All right, we're going to go through 3 different types of pathways. The first 2 are quite complex. I've simplified it as much as I can. And the third one's really easy, so you can look forward to that. The first type of pathway that I want to discuss is something called an RTK, or receptor tyrosine kinase. Now I'll discuss what these parts actually mean in a minute. You can refer back to the slide if you want a definition. But let's take a look at a diagram. First I need to tell you about the functions of RTKs before we get into the actual steps. All right. So let's say that you wanted to send a signal to a cell and tell it to do something complicated. Like if you said to a cell, divide. Well, division is a really complicated process. In order for a cell to divide, it has to change its DNA, it has to break down the nucleus, it has to activate things like centrioles, it has to move organelles around. There are a lot of different jobs that have to be accomplished in order for the cell to divide. But it's still possible for you to send it a signal like divide, a single word, and initiate all these different processes. And that's what RTK pathways do. A single signal has many effects inside the cell. Now let's take a look at how it works. The receptors in an RTK pathway are kind of unique because they're actually in 2 parts. There's part 1 and there's part 2. When the receptor binds to the ligand, and one will have to bind on each side, it causes the 2 parts to come together, or dimerize, and form a dimer with 2 parts, di for 2. Once the thing is dimerized, it's ready to go. Now notice, on the outside of this thing, there's some amino acids sticking out, and these things are called tyrosines, and it's got a bunch of them. That's why it's called a receptor tyrosine kinase pathway. Attached to this protein, there's also going to be something called a kinase. That's what the K stands for, is the kinase. And what it does is it's going to take some ATPs and strip phosphates off of them, making them into, I can't circle very well, but making them into ADPs. And it will take the phosphates off those ATPs and stick them onto the tyrosines. So these tyrosines are being phosphorylated. And if you remember, those phosphates represent energy. So this molecule is now energized and ready to do some work, ready to go. What will it do next? Well, the energized form of this molecule will now interact with these things, which are very generically called relay proteins. They're proteins that are going to get activated, they're going to travel deeper into the cell, and they're going to do some kind of work, or activate another kind of protein. So, ligand binds with receptor, it dimerizes, it adds phosphates to itself, that activates relay proteins. And these relay proteins can activate a whole lot of other things inside the cell, so it can do something complicated. All right. Now the second kind of signal transduction pathway is known as a GPCR, or a G protein coupled receptor. These types of pathways tend to be associated with a single response. So this kind of pathway isn't very handy for doing something like, say, division, a single message that causes something complicated to happen. This type of pathway is used more for one specific task. Release chemical A, something like that. Let's look at the functions of some GPCRs. All right. So a great example of where GPCRs work in your body are in the sense organs. And we're going to look at the sense of smell. Now, inside your nose, way up here at the very tippy top, right under your brain, there's a little patch of tissue here called the nasal epithelium. And if we look inside that, you're going to see these yellow-looking cells sticking out. Those are neurons, and they're going to receive information. They have these little extensions that stick out, and those are going to have receptors on them for smell molecules. And of course, you know that when you smell a chemical, little particles of that chemical go up into your nose. They're going to touch these receptors, and that's going to cause a signal transduction pathway that travels to the brain. So let's say that you smell something like popcorn. Popcorn molecules, the aromatic parts, go up and they bind to the receptors. And there's going to be a single G protein pathway connected to the smell of popcorn, specific for that. That's going to tell your brain, hey, I smell popcorn. If you smell smoke, that's going to have a different G protein pathway associated with it, so that you can send a very specific message to the brain. So G protein pathways tend to be very specific for specific functions. Now, what are the steps of the pathway? How do they work? Well, let's go through it by looking at a diagram. As always, this pathway is going to start with a ligand and a receptor. So the ligand binds with the receptor. Now that the receptor is going to be attached to the special kind of protein called a G protein. There it is down there. And when it changes its shape, it's going to activate the G protein. That G protein is going to take off inside the cell, and it's going to travel to this special molecule called adenylyl cyclase. That's it right there. That's messy, but you get it. Now, what does this crazy guy do? Well, what it does is it takes ATP and breaks it down into something called CAMP. What is that about? What does that mean? Well, you know that if you take ATP and you remove a phosphate, if you dephosphorylate it, you get ADP, adenosine diphosphate. If you were to take off an additional phosphate, one more, you'd be down to just one, and that's adenosine monophosphate, right? Just for kicks, let's take a look at that little molecule, because we haven't seen it before. This is CAMP, or cyclic AMP, also known as CAMP, and it is related to ATP. It's a nucleic acid, so it's got a sugar, it's got an adenine, there's its base, and here's its phosphate group. Because it only has one phosphate instead of three, it kind of loops around on itself and it makes this little loop. So that's why it's called cyclic AMP. But that's what it kind of looks like. And cyclic AMP is known as a secondary messenger in these pathways. So it kind of picks up the message and carries it further into the cell. When levels of CAMP begin to rise inside a cell, that tells the cell to do something specific. All right, so in that G protein pathway, the adenylyl cyclase makes the CAMP, those levels rise, you get a response. Finally, we're going to talk about ligand-gated ion channels. This is the easiest of the pathways, so we'll end with that. You guys know what ion channels are already, but let's take a look at them just to remind you. A channel is just a tube that sticks through a membrane, and it allows certain kinds of molecules to pass through. Some channels are capable of opening and closing. And so in this case, the opening and closing is being controlled by this little ligand. When this signaling molecule comes along and binds with this protein, it opens, and that allows something to go into the cell. When the ligand detaches, it closes. These types of ion channels tend to be specific as to how they function. So some open when they bind to ligands, others close when they bind to ligands. And they're all specific as to what they allow to pass through. That can differ as well. But that's the whole pathway. That's really easy. Open, close. All right. Now there are a couple of more questions on that quiz that I created. These last two are brain burners. You're going to have to really analyze these pathways and really think about this before you make your guesses on these two. And I picked them out specifically because they are difficult. I won't put any that are quite this difficult on the test, but I wanted you to practice with these. All right, and you can always consult with your neighbors on these things as well, see what they see. All right, so there's the first one. Here is the second one, talking about cholera, which is kind of interesting. All right, so that is it for chapter 11. I hope this lecture was helpful. If you find this topic particularly confusing, come and talk to me about it. We can work through whatever problems you're having with it. And before you walk away from this chapter, I would like you to complete this quiz. This quiz is for a grade, and we're doing it outside of class instead of in class this time. So again, what you need to do is either go to your ACC email and look for an invitation from me to open this quiz, or you can click on this link down here at the bottom or type that in, and that should take you directly to this quiz. I created it in Google Forms. It should grade it automatically for you and show you what you got right and what you got wrong and give you feedback on those questions as well. So that should help clarify what the correct answers are and why. All right, so best of luck with that quiz. Make sure you get it done within the time limit. I will specify that via email. And I hope this was helpful to you.